we focused on the detection of cervical cancer cells from pap smear images using their texture features. So cervical cancer is one of the most prevalent forms of cancer within women. There is an estimate of 14,000 women that are diagnosed with cervical cancer each year, but in its early stages, it could be highly treatable. They are able to determine whether or not an individual has cervical cancer through a method called a pap smear test. And in this on the detection of cervical cancer cells from pap smear images using their texture features. Cancers are cervical cells consisting of both normal and abnormal. On the bottom left, you can see that we first simulated all normal cells to kind of get a baseline and a control for those values that we should see for just normal cells. And you can see that the correlation is around 0.93, the energy is around 0.97. And as you add those precancerous cells to the layouts within our simulated environment, you could see that there are certain trends that will follow and do follow within the analysis of 144 cervical cells. And on the right, you can see those cervical cells as well being analyzed with the same amount of trends that are forming within the data set. Speaking about these trends, you can see them here in this visual representation. On the left, you see the analysis of the 36 cervical cells. Their first analysis we did was the 36.
This project was done in collaboration between the New York Institute of Technology and members of Virginia Tech. Just to provide some background, organosulfur compounds, otherwise known as OSCs, are characterized by one or more sulfur atoms bound to a cyanide group or a carbon atom in a cyclic or non-cyclic fashion. These molecules actually have an extraordinary range of chemical structures and activities. Many of these activities include anti-cancer properties, and recently there have also been reports of antibacterial activity. Examples of OSCs are included below, which are diallyl sulfide, diallyl disulfide, and allicin. Allicin is actually found in garlic, and it is one of the OSCs that may be responsible for the anti-cancerous and antibacterial properties of garlic. In addition, other examples are E-adrine and benzyl isocyanate. An important aspect of OSCs is that they may release H2S, which may serve as a signaling molecule in a variety of cell types, including mammalian cells and bacterial cells. So H2S can modulate protein diffusion assay. 3-MPITC inhibited bacteria in a concentration-dependent manner in E. coli and
Hi, this is Patricia again. I do have another question for a different speaker, Zaman, the one who talked about the organosulfur compound. You did mention that you encountered some solubility issues. Can you expound more on what sort of solubility issues you encountered with the
Sure, yeah.
organosulfur compounds that you
Yeah, thank you for the question.
at.
So, the OCs that we tested are oily compounds, and we were attempting to dissolve them in DMSO. So it seemed that while we were conducting the H2S release studies, the compounds tended to settle towards the bottom or aggregate. We do plan on addressing this issue by perhaps dissolving the compounds in more DMSO, which should help mitigate those effects.
Okay, and a somewhat related question. You did measure H2S that's evolved. Can you tell me a little bit more on how you guys measure this? Since H2S is a gas, yes. Thank you.
So, as mentioned before, so we use methylene blue as an indicator. So, how that works is that once H2S is released, it acts with a compound within the reaction, and all it does is like it produces the methylene blue as a result. So that's like the basis of the assay.
Again, the determination of the minimal inhibitory concentration.
Sure. So we tried different concentrations of the bacteria of the compounds. On the disc, the concentrations were much higher, so they were ranging from 750 millimolars all the way down to 38 millimolars. And what we did was, towards the lower end of those values, we saw that there was no zone of inhibition. So as soon as we saw a zone of inhibition, which tended to be 8 millimeters, that's when we determined that that was the minimum inhibitory concentration. And I would just like to say that the size of the discs were 6 millimeter, just for comparison.
Okay, thank you.
Thank you.
I have a question. Would you be suggesting any statistical analysis to analyze the results of your image analysis, of your image?
Yes, there's multiple forms of quantitative analysis that we can use to basically determine the accuracy of these tests. We actually noticed from the p-value of the energy that it was within the range to be considered significant. In addition to that, when we do perform these tests, if we perform these tests again in the future, if we have a larger data set, we will be able to perform that quantitative analysis and have those results to see the accuracy. We are also planning on putting this into a deep neural network, which will also be able to assess those quantitative forms of analysis and measurement.
Rahman, you mentioned about the degradation of organosulfur compounds over time, and that can impact with your analysis. How do you plan to take care of that moving forward?
Yeah, so moving forward, first we would like to correct the solubility issues so that we can better resolve the differences in H2S release between the different compounds. Additionally, we would like to conduct mass spectrometry because currently, we did analyze the compounds based on their structure on a theoretical level. Doing mass spectrometry would allow us to see which products are actually being produced. And I believe that would be important because, as you can see in the results, 3-MPITC was more effective against the bacteria than DFFDS. But in the H2S release assay, it's seen that DFFDS release more H2S than 3-MPITC. So there may be other products in play here, so we would just like to confirm those. And thank you for the question.
Thank you.
Question for Donnel about in your study, so you developed your methodology using the training images. So you have very kind of uniformly distributed cells that you were then identifying the abnormal cells. But then obviously within the clinical samples, these are going to be much more complex and heterogeneous. And so I was wondering how your methodology kind of takes that into account.
That's a great question. So when actually developing the threshold that we use to determine whether or not it was abnormal or normal, we took multiple features into account. The entropy was just the most significant feature. There are also minor changes in homogeneity and contrast and correlation. And one of the other things is that we tried it for multiple different cell staining and cell preparation methods to account for the differences that occur in different samples that are acquired from a lab. In addition to this, when we are putting it through the algorithm, we compensate by a significant amount for that amount of error. In the graphs that I put as well, there were some error bars for the bireal data set that was tested, and it shows that the values are within a certain range when those abnormal cells are present. As for other differences, such as maybe a darker feature that is within the threshold that is not the nuclei of the cell or other cytoplasmic features, the threshold level can be improved through future testing by fine-tuning exactly which values correspond to the abnormal cells and the normal cells, and the threshold, their masking can be trained so that it does not pick up any of those other features within the image that are not part of that.
Because I don't know anything about how pap smear samples are analyzed, but are there different methodologies that are used currently by the people who are analyzing these visually? So different kinds of staining protocols that are used, or is it kind of standardized already?
Well, as for the cell staining method, it's particular to the cytopathologist that's doing the screening for it. One of the beautiful things about our algorithm that we use is that other than contrast, the contrast of the grayscale image, the actual cell staining method doesn't affect these other values significantly. So although they might use a darker staining method or a lighter one, those texture values are consistent throughout the analysis. And when they were tested, when different abnormal cells that were prepared differently were tested, they all fell within this range of below about 0.88 of an energy value, with the corresponding correlation and homogeneity values for that specific subset of abnormal cells. But as time progresses and we are able to have a larger data set, those will be fine-tuned.